Now let's discuss about the pathogenesis. So what are the mechanisms of cell injury? Mechanism includes, first one is hypoxia and ATP depletion. Hypoxia resulted in abnormal metabolism that is associated with the following changes that are reduced phosphorylation, hence ATP, and then increased anaerobic glycolysis, increased lactic acidosis, decreased pH, that is cellular acidosis. And the sequence of the above mentioned disorders include clumping of chromatin, loss of ribosome, and then intracellular influx of water. Now coming to the second mechanism, we have the formation of free radicals like superoxide, hydrogen peroxide, hydroxile group. Those leads to damage of cell membrane with the loss of sodium, potassium, and influx of calcium ions. Energy depending sodium pump also slows down. So the normal activity of the sodium pump keeps the intracellular concentration of potassium significantly high. So the failure of active transport through the cell membrane causes accumulation of sodium and water within the cell and potassium out of the cell. So this is the mechanism we have. So because of the, I repeat again, because of the failure of active transport through the cell membrane causes accumulation of sodium and water within the cell and potassium out of the cell. Thirdly, we have the increase of intracellular calcium. So the release of free calcium ions from intracellular stores with the activation of different enzymes. So ischemia and hypoxia are the most common forms of cell injury. Now coming to the pathogenesis, let's discuss about let's discuss about the reversible cell injury. So if the ischemia or hypoxia is of short duration, the effects may be reversible or rapid restoration of circulation, example in coronary artery occlusion, myocardial contractibility, metabolism of ultrastructure are reversed if the circulation is quickly restored. So reversible injury denotes pathologic changes that can be reversed when the stimulus is removed and the cellular injury has mild. So cell injury is reversible only up to a certain point, otherwise it will be irreversible. Now let's look into the changes in reversible cell injury. First, we have the cellular swelling. Due to the accumulation of intracellular water and the swelling of cisternae of the ER, endoplasmic reticulum, it will result in, first, loss of microvilli, second, Blebs formation, third, swelling of ER and mitochondria, and clumping of chromatin. So here is a picture. We have the clumping of chromatin, ER swelling, membranous Blebs, inclusions, mitochondrial swelling. So these are the cellular changes. Now coming to irreversible cell injury, it denotes pathologic changes that are permanent and causes cell death. They cannot be reversed to normal state. For example, if the blood supply to the heart muscle is cut off for 10 to 15 minutes, then the process will be halted or it will not be proper. So however, if the blood flow is cut off for a long period, then the myocardial fiber dies and then necrosis occurs. And then irreversible injury is marked by severe mitochondrial vacuolization and then density formation, extensive damage to plasma membranes, detachment of ribosomes from the granular endoplasmic reticulum, etc. And the injury to lysosomal bodies leads to a leakage of lysosomal enzymes in the cytoplasm. And there is no universal agreement to biochemical change at the point of no return. So here's a big picture. So let's discuss about the events that takes place in irreversible cell injury. First, we have the ADP depletion. So as a result of mitochondrial dysfunction, that is because of lack of oxidative phosphorylation, ATP is depleted and then production of energy is reduced. Second, we have cell membrane damage. The earliest phase of irreversible injury is associated with the functional and structural defects in the cell membrane, and a great deal of evidence indicates that cell membrane damage is a central factor in the pathogenesis of irreversible injury. Intact cell membranes are essential to the maintenance of normal cell permeability and volume. Loss of membrane integrity causes massive influx of calcium from the extracellular space that results in mitochondrial dysfunction. Activation of intracellular enzymes and denaturation of proteins, etc. includes in cell membrane damage, and the persistence of ischemia or hypoxia result in reversible damage to the structure and function of cell death, and the stage at which this point of no return is irreversibility is reached from reversible to a reversible cell injury is unclear, but the sequence of events is a continuation of reversibly injured cell. So that's all about the pathogenesis in brief. So let's go into the summary of what we've discussed.